Hi, my name is Anna Quiroga. I'm CEO and co-founder of SANICKA, a Georgia Tech and Emory University spin-out incorporated in Georgia. In SANICKA, we believe that every cancer patient should receive optimal treatment. More than 60,000 people are diagnosed with leukemia in the US every year, and most of these patients are going to receive chemotherapy, a treatment that has saved millions of lives, but is associated with some risks due to the one-size-fits-all approach to select treatment. Some of these risks are underdosage, that means the treatment is not going to be effective enough, overdosage, that means the patient is going to be exposed to the treatment side effects, and resistance. 20% of patients are resistant to chemotherapy and they receive a toxic treatment that is ineffective. So for this reason, we developed PICIT, a precision chemoimmunotherapy tool that can predict and simulate how the patient and tumor are going to respond to different treatments. And then we can select the best treatment for that patient and their tumor. And also we can optimize the drug, the dose, and the schedule to improve outcomes and reduce toxicity. So these are some results from 2 patients that received intensive chemotherapy. One of them achieved complete remission and the other one unfortunately relapsed. Here we can see the percentage of leukemia in the bone marrow. The circles are the patient data, the crosses are the simulation results, and the arrows indicate when the treatment started. We can also see the absolute count of neutrophils in peripheral blood that are measured daily. Those are the dots, the patient results, and the lines are the simulations from PICIT. So the results from our validation with patient data indicate that we can predict the overall outcome for all patients, so complete remission, partial remission, relapse, and resistant disease. We can also predict the percentage of leukemic cells in the bone marrow for 91% of the bone marrow assessments. And also, we can track the neutrophils count for 89% of patients. So how does it work? The hospital can upload the data into the cloud using the website, right? And SANICKA collects that data and performs simulations and optimizations using our mathematical mechanistic model. And we provide clinicians with visualizations of how the patient is going to evolve and also the disease. And the clinician and patient together can decide which is the best treatment. The patient is going to receive this treatment in the hospital where it's going to be monitored. And then hopefully we can achieve disease control or cure with lower toxicity, faster recovery, and higher efficacy. So PICIT provides hospital with predictions of patient response to different treatments for treatment selection, and also optimization of drug, dose, and schedule to improve outcomes and reduce complications. So today, we could potentially help 20,000 people with acute myeloid leukemia, reducing complications and improving their outcomes. And also, we could save around $100,000 per patient per course of treatment, and also help the healthcare system to save around $2.2 billion. We are focused now in acute myeloid leukemia patients, but we are going to expand to all leukemias in the US and then hopefully reach the 8 major markets. Our revenue stream is focused on hospitals, in particular NCI centers, where they can perform the tumor characterization. The revenue model is per patient per course of treatment. That means that we are offering to simulate 1 to 6 cycles of treatment, and after that, we can simulate how the patient is going to evolve after treatment to predict if the patient is going to relapse and alert clinicians. Our reimbursement strategy is to apply for the new technology add-on before we get the diagnosis-related group coding. So today, the challenges in the standard of care are first personalization, then also tumor specificity, and also the incorporation of novel therapies that are reaching the market. And finally, predict the dynamic evolution of the disease and also the patient recovery. PICE-AT can do that. PICE-AT is personalized, tumor-specific, and we can incorporate all the drugs that are coming into market based on the mechanism of action. And we can also predict the disease evolution and the patient recovery. There are no competitors in the market. There is one competitor that is about to reach the market. They are still recruiting for clinical trials, but they are very tumor specific, and they can classify patients as responder or non-responder to certain therapies, and they cannot predict how the patient is going to evolve. In our path to commercialization, everything started more than 15 years ago. The BSCL laboratory at Imperial College London was studying blood production and also blood disease disorders. And then we moved in 2018 to the US, and we incorporated this year in 2021. And we got the CreateX grant and also the Georgia Research Alliance grant to validate the pediatric algorithm. And also we got the Biolocity grant to build our minimum viable product and test it. And we are also applying for the NCI Concept Award for the MVP for pediatrics and the Fast Track STTR for the clinical validation of PICE-AT. We are planning to launch PICE-AT simulator in 3 years. It doesn't need FDA clearance, but PICE-AT or PICE-AT Optimizer requires FDA clearance because it's proposing a non-standard treatment that is personalized. Our next steps are, well, first, include all the FDA-approved drugs for acute myeloid leukemia, and also include tumor characterization with single-cell multi-omics data, and the validation, observational and prospective with our collaborators. Our team, well, there is Dr. Mantzelaris, he's professor at Georgia Tech, Dr. Panoskalsis, she's an hematologist at Emory University and also an associate professor, and myself, I'm a postdoctorate fellow at Georgia Tech. We have the support of Georgia Tech and Emory and the Children's Hospital, and also from all the support from our mentors from the Georgia Tech Venture Lab, CreateX, Biolocity, GRA, and ATDC. Please join us and help us to put PICE-AT in the hands of oncologists.